Section A, and this is one of the last two for this unit. First, you would need to think about the difference between eukaryotes and prokaryotes, or eukaryotic and prokaryotic cells. Again, this is background information, but some of the differences I'll write out for you. Eukaryotic tend to be multicelled or multicellular, and prokaryotic tend to be unicellular, which automatically will make them smaller. Eukaryotic tend to have membrane-bound organelles. If you think back to biology classes, when you talked about the endoplasmic reticulum and the nucleus, everything had its own membrane, whereas prokaryotic cells are non-membrane-bound organelles. In general, eukaryotic are much larger, prokaryotic are much smaller. Eukaryotic are also have a slower metabolism. They're simply larger, and you have to figure that there are more membranes to get through and everything is occurring within the cell. And prokaryotic have a much faster metabolism. Consider that the size is also much smaller and things are not membrane bound. So how do bacteria replicate themselves? Bacteria are asexual, and they replicate themselves using a process known as pretty much binary fission. This is a large majority of bacteria. So notice there is genetic material inside, but it is not membrane bound. And then they start pulling apart, and then there is a pinching off process, and you end up with two daughter cells at the end of binary fission. On the other side of the screen, you can see budding. This is also an asexual reproductive process and what most fungi do. So it's similar in the sense that you have your genetic material, and then they start separating. Notice here a spore or forespore is formed, and inside you have a spore coat. This allows fungi to handle more extreme conditions, and then it sort of buds off within itself, which is why it's known as budding. We need to talk about cell morphology, because how we actually define bacteria. Remember from previously we said it can't be phenotypically, because simply looking at a tube, even the colors and everything else is very simple. So we look at the 16S rRNA. Another factor in determining bacteria and identifying them is their shape. Morphology, another name for that word, is in fact shape. So the main ones are the various shapes. We have coccus or cocci. These are the round ones. So that's one main one. A second main one is bacillus, also known as bacilli. These are rod-shaped. And finally, we have spirillum, which are essentially spiral-shaped. You can actually see these. Here you see the rod-shaped bacilli, and here you see the round cocci, and this bottom picture here is the spirillum. There's a statement on the bottom that's a little bit important. Old cultures tend to exhibit pleomorphic characteristics, and it is asking why. Pleomorphic, pleo means many, and morph is the shape. So when a culture is old, they tend to look different shapes, and they don't look properly. Imagine a tube that has broth in it, which is media or the liquid that you're already learning about in lab. Inside of here, of this broth, you have the oxygen that the bacteria requires, and you have the nutrients. As this bacteria grows, it eventually is using up all of those nutrients and all of that oxygen. It is defecating, urinating, doing all of its, so to speak, bodily functions within this same tube. This is almost like us laying into a bathtub and eating and defecating and doing everything in one place. Eventually, the waste products overtake the needs of the oxygen, the nutrients, and vitamins, and everything else that's in the broth. When a culture tends to be old, the waste products go up, which causes bacteria to shrivel and change shape. So when you're doing microscopy work in microbiology labs, you always want to use new and fresh cultures, because when they're old, they tend to change shapes. And the why is because the oxygen and the nutrients are essentially running out. So if we take a look at this, and I add one more aspect to this, we just covered morphology, but there is another term that's incredibly important, which is known as arrangement. There are basically two types of arrangements that we're going to cover in here. One is staff flow, which are the arrangement or shape of what would look like clusters of grapes. And the second is strepto. These are actually chains. When you name a bacteria, the order is first you name the arrangement, and second you name the morphology. So if you take a look at this picture here on the bottom right, you have an arrangement of chains. So this has to be strepto. And then notice those are rods. The term for rods is bacilli. So this particular picture represents streptobacilli. Here up top, if you look at the arrangement first, it's also chains. So this is strepto, and these are little round balls. So it's streptococci. Remember that first you name arrangement and next you name morphology when you're looking at the shape and the arrangement of bacteria. If you look at this picture on the left, the round ones are cocci and these are spirillum. We focus purely only on staff flow and strepto. So if you look right here, these are essentially chains, which make them strepto, and they're chains of rods, so they're streptobacilli. There are some unique terms that are on here for practice, so I can discuss them with you, but I do not test you on them. So instead of calling these streptobacilli, if you were working in a research lab, you would call this diplobacilli because there's two of them. This would also be a diplobacilli down here. So let me give you one more example. If you have an organism and I have you draw it, and it's staff flow in arrangement and cocci or coccus in shape. Staff flow means clusters. Cocci are round little balls. So staphylococcus would look this way, like a cluster of grapes. If we had something that was strepto in arrangement and coccus in shape, strepto means chains, so it would be a chain of round little cocci. Now we're going to start covering all of the bacterial structures. So hopefully you have your outline printed, and this begins on the bottom of page 3, and this information is going to wrap up the end of Unit 1. Please remember, as I went through it in your Unit 1 lecture outline review, that for all of these structures, and there's 11 or 12 of them, you want to know the composition, which is it made of carbs, lipid, protein, or nucleic acids? Is it found in gram-positive or gram-negative bacteria? What's its function, and is the structure some type of virulence factor, meaning does it increase the chance of infectivity and allow the bacteria pathogenically to cause infection? So first we're going to look at pili. Pili are essentially little appendages hanging off of the edges of the bacteria. Their composition is that they are made of proteins, and they are found in both gram-positive and negative cells. Their jobs have to do with not only transferring DNA and certain types of motility, but their actual functionality is that of attachment. So they have something known as adhesins, that's sticky factors. Think of when children in a gumball machine pull out those little sticky hands and you smack it against the wall. Pili function in the same way. They attach to things that they come in contact with and come close to. If you take a look at the picture on the right, you have two different bacteria and you have a pili that managed to attach itself between the two different bacteria, this is a means in which DNA can transfer back and forth, causing various mutations. It's called horizontal gene transfer, and we're going to cover this in full detail in Unit 2. I'll also mention flagella. Flagella are the tails at the end of bacteria. These are made of protein and also found in both gram-positive and gram-negative. What do? They provide motility, which is the ability for the bacteria to move. This type of motility is known as chemotaxis. And there are two different kinds. There is a positive chemotaxis, that there is, say, a nutrient source, and if it's a positive chemotaxis movement, the bacteria is moving towards that signal. If it's negative chemotaxis, it is moving away from that particular signal. So positive towards an attractant, negative away from a repellent. If I were to draw a bacteria down here, and here is its flagella, and I wanted to show an example of a positive chemotaxis, I would put that positive here because the tail or flagella is always behind it, and this implies that the bacteria is swimming towards or moving towards or completing chemotaxis towards that positive signal. If, on the other hand, it's a negative chemotactic signal, I would place it on the opposite side, because that shows that the tail area is behind the bacteria, and again, it is swimming away from that negative signal. A little explanation of virulence factors is this is a physical molecule or some type of structure that a bacteria has that provides the ability for it to be virulent or cause disease. In this example, the pili does have a virulence factor, and that virulence factor is called adhesin. Adhesin has a sticky property, and that in the previous picture is what allowed it to stick to something. If you look at this picture on the left, here's your host cell, and this is a bacteria. All of these little yellow appendages are pili, and it is attaching to receptors on the host cell for the sake of being a virulence factor and providing it the ability to be infectious. Here are a couple more pictures that's showing adhesins, a little more detailed, but just so you can see it again. So receptors are molecules on the surface of the cell, which are embedded inside of the membrane. When you have an adhesin, which is the virulence factor of the pili we just discussed, the pili lock into those receptors, giving it that virulence factor and ability to be infectious to its host. Next, we're going to cover a structure known as the glycocalyx, and this one is going to also relate to an entire short answer, almost essay question, 5 or 6 points on the exam. So please make sure that you understand and remember the key relevant points as they relate to it. The outer layer of a membrane, if you take a look at it, this orange one right here, so this orange one right here is actually the plasma membrane, which we've talked about previously, but we'll bring up again. Then the purple, or the next one, is the cell wall, which is also coming up. But the pink, this pink outermost layer is known as a glycocalyx. Glyco implies sugar, so this is actually a polysaccharide. That is its component. I'll write it here for you. And again, this is all on your outline, so please make sure you have the outlines at least printed. So this is a polysaccharide, but it has two other names, capsule and slime layer. All three mean the same thing. I'm going to call it glycocalyx and capsule back and forth so that everybody remembers. If you have a dog and you clean the water bowl, you feel that slime on the bottom. Or if someone notices the bottom of a contact lens, if you wear contact lenses, and there is that sliminess on the bottom, what you're feeling is the glycocalyx from bacterial buildup. If you look at the picture on the right, the little black portions right here in the middle are all of the bacteria, and this grayed out zone area around the bacteria is actually the glycocalyx, and the capsule, and the slime layer, all three of them mean the same thing. So what exactly does it do? The glycocalyx is found, or I'll just write capsule here so that you get used to hearing all three. I've already mentioned that it's a polysaccharide, so it's made of many sugars. It's found in both positive and negative gram cells, and it's got a lot of functionalities. One of them is to prevent dehydration or desiccation. It also attaches for the sticky factor. So the virulence factor here is the fact that it is so very sticky. Think of a contact lens or a dog bowl, the bottom of a water bowl for a dog, where you can feel that softness. So what it does is it gives essentially power in numbers to bacteria. It allows them to stick together on surfaces, which gives them the power of being a virulence factor, which then allows it to attach easier to its host as well as cause disease. It also stops phagocytosis, which is the host's immune system, in breaking down the bacteria that's trying to invade the body. I want to talk a little bit about biofilms because, as I mentioned, that is an aspect that's going to come up a lot. Biofilm is what you have or what you get because of the sticky factor of the glycocalyx. It's made from excessive glycocalyx. So what does it do? It provides a virulence factor, which we've talked about through its sticky factor. It gives power in numbers and protection to the bacteria to allow it to not be killed off by the host's immune system. But one thing it doesn't do, and this is important to remember, is that it does not provide nutrients. Why is that important? Because traditionally, when we talk about sugar and glucose and things of that nature, we always assume that it's a nutrient source. In this case, the glycocalyx is not a nutrient source. It's purely for the virulence factor of being able to stick together. So understand the advantages of being a virulence factor and power in numbers. Know a couple of examples. There are ones in the allied health field, such as a catheter that has biofilm buildup, which is why it needs to be changed in the patient often enough. And more simple thoughts are the water bowl for the dog, and again, the contact lens one. If you take a look at this picture, this is a biofilm that is seen in a contact lens. So what you're looking at is all of this aggregate bacteria sticking itself together by the glycocalyx sticking together. This is a virulence factor, which is why you can impact your vision negatively if you don't clean your contact lens cases or keep them in a proper cleanly manner. You do have to change them because they're plastic every so often. The picture on the right really nicely aligns with allied health. The main one is a catheter that I'm going to use in an example, but if you think of a respiratory tech or a trach, for example, anything entering the body has the ability to have issues with glycocalyx buildup, also known as a biofilm. If you look at the top picture, the urethra, male or female, where you insert a catheter, is always going to have yeast and other bacteria. That area is very not sterile. But if you take a look at the top picture versus the bottom, in only 24 hours after the insertion of a catheter, look at the incredible amount of growth, which is why it does in fact need to be changed on a regular basis to keep the patient safe and healthy.